Hi guys, welcome back to PathDemystified. So today's short tutorial is going to be about synthesis of thyroid hormones. I'm Dr. Shubhika Sachdeva and let's just dive into the topic. So do you know where thyroid is located? Thyroid gland is located in anterior of your neck, just inferior to your larynx, that is your voice box. And this is a butterfly shaped organ and it is one of the largest endocrine glands in our body. This gland is responsible for secreting three main hormones in our body. The first two are the metabolic hormones, which increase the basal metabolic rate of our body, and those are thyroxine and triiodothyronine. And the third is the hormone which regulates the calcium metabolism in our body, and this is secreted by the C cells in the thyroid, and this hormone is called as calcitonin. Today we are going to concentrate ourselves on the synthesis of the two basic metabolic hormones, that is the thyroxine and triiodothyronine. So, thyroid gland consists of something called as thyroid follicles. These thyroid follicles are the structural and the functional unit of thyroid gland. Can you see here? These are individual thyroid follicles. Each thyroid follicle is lined by simple cuboidal epithelial cells, and the lumen of each follicle consists of something called as colloid. This colloid consists of a glycoprotein called as thyroglobulin. We'll understand the concept of thyroglobulin in subsequent slides. Now, can you see this triangle shaped cell just above the thyroid follicle? These are your C cells, and these cells are what which secrete calcitonin. Now, understanding it microscopically. First, see here. This is the capsule. Thyroid is an encapsulated organ. So this capsule, thin capsule that you can see outside in this pictorial picture, is what you can see microscopically here. And all this here are your thyroid follicles arranged back to back. Now let us zoom in, let's say on one of the thyroid follicles here. So once we zoom in the thyroid follicle, you see this. One second, I'll just erase this for you to make it easy. Yeah, so now you see this is one thyroid follicle which is lined by simple cuboidal epithelial cell. Can you see this blue cells? These are your cuboidal epithelial cells. This homogeneous pink material inside the thyroid follicle is what is known as colloid. The space between the two follicles, this is one follicle, this is an other follicle, this is an other follicle, this is an other follicle. So the space between the two follicles is what we call as interstitium, which contains your red blood cells. Okay. So we are going to study about the steps of thyroid synthesis in a while. Let us first understand that where does this all thyroid hormone synthesis actually begin. So first of all, it begins at the level of hypothalamus. This is your hypothalamus. This is your anterior pituitary. Okay. There are specific neurons or nuclei at the level of hypothalamus which are called as the paraventricular nuclei. These paraventricular nuclei are responsible for secreting a specific hormone called as the TRH, that is your thyrotropin releasing hormone. This TRH, once secreted from the hypothalamus, will travel through the vascular system to the anterior pituitary and stimulate specific cells on the anterior pituitary called as the thyrotropes. Once these thyrotropes are stimulated by the TRH, this anterior pituitary will now release a specific hormone, and a very important hormone called as the TSH, standing for thyroid stimulating hormone. This thyroid stimulating hormone will now travel through your blood to the thyroid, and to the thyroid follicle. So there are two important hormones, that is, first is your TRH released from the hypothalamus, which goes from the through the hypophyseal portal system into your anterior pituitary here, stimulates thyrotropes in the anterior pituitary. You see these yellow cells, these are all thyrotropes. They stimulates thyrotropes and releases a hormone called as TSH. This TSH will now travel through the blood to the thyroid gland. So once your TSH is released from the anterior pituitary, it will circulate in the blood and reach your thyroid gland. So let us just zoom in into one of the thyroid follicle. Let's say this is a zoomed in picture of a thyroid follicle. Now to understand that, this TSH coming from the anterior pituitary will now go and bind on the thyroid follicle to its receptor called as the TSH receptor. Now, this TSH is a peptide hormone. What does that mean? That means that it will bind to the receptor present on the cell membrane and not inside the cell. So, once this TSH gets bind to the TSH receptor on the thyroid follicle, it will stimulate intracellular pathway, specifically a G stimulatory protein. Now, normally this G stimulatory protein is binded to GDP, which keeps it inactivated. Once this binding of TSH with TSH receptor has occurred, this G stimulatory protein gets binded to GTP and it gets activated. Once this G stimulatory protein is activated, it will go and activate a effector enzyme called as adenylate cyclase. Once this effector enzyme is activated, now what happens? This effector enzyme converts your ATP into CAMP, and the increased levels of CAMP will lead to the production of an enzyme called as protein kinase A. Now, very interestingly, when this protein kinase A increases, what will it do? It will go inside the cell and it will phosphorylate the transcription factors present on the DNA. Once the transcription factors are phosphorylated, it will go and activate genes on the DNA and give them signal to produce certain proteins, and it will activate a cascade of transcription. So once the genes are activated, it will lead to the formation of mRNA. This mRNA will go to the rough endoplasmic reticulum and make amino acids. These amino acids will undergo translation and produce a protein. Once this protein is produced, it will now go to the Golgi apparatus and get packed. This Golgi packs the protein in a vesicle. Now see, this vesicle, this is a vesicle, and inside this we have our protein, okay? So now this vesicle is a lipid bilayer, and this membrane is also a lipid bilayer. So these two lipid bilayer will fuse with each other, and once they get fused with each other, they will release the protein into this luminal space. See, this protein is here, it is getting released into the luminal space. Now this leads to the formation of the basic protein for thyroid hormone synthesis, which is called as thyroglobulin. Okay. So once we have understood the formation of the basic unit of thyroid hormone synthesis, that is the thyroglobulin, we need a few other things for this reaction to take place. So we need iodine. Where do you get this iodine from? So we get this iodine from our diet. Various nutrient sources or iodinized salt that we eat on daily basis is what provides this iodine. Now this iodine is present in form of an iodide ion in our blood, that is, it has a negative charge to it. Now this iodide would require to go inside the thyroid follicle cell, correct? How does it do that? So what we need to understand is that the iodide concentration is less inside the blood as compared to the thyroid follicle, correct? So for something to go from a low concentration to the area of high concentration, we require energy. Correct? So where do we get this energy from? Thankfully, we have a good old friend, sodium, to help us out. Sodium is present in higher concentration inside the blood as compared to the thyroid follicle, right? So this sodium will go from blood into the thyroid follicle, and along with it, it will take one molecule of iodide with it. So when it's something is sodium is taking something with it, it is called as a symporter or a co-transporter. Now this sodium iodide symporter is using ATP indirectly from the sodium potassium ATPase pump. So when it is using ATP indirectly, this kind of transporter is called as a secondary active transporter, right? So once this iodide is inside the thyroid follicle, it would want, okay, before that, the concentration of iodide inside the thyroid follicle is what we call as iodine trapping. So this is another step in thyroid hormone synthesis, that is iodine trapping. Now this iodide is, once it is inside the thyroid follicle, it would want to go into the luminal side. So how does this happen? We have another protein called as pendrin. What is pendrin? It is the chloride iodide ion counter transporter. What does that mean? That means that it will pump the iodide ion into the luminal space of the thyroid follicle and will take one ion of chloride from the luminal space outside. So it is a chloride iodide ion counter transporter. Okay. So now once this iodide is inside the luminal space in the thyroid follicle, we would want to convert it into iodine. So how does that happen? Now we have a very interesting enzyme called as, one second. So now we have a very interesting enzyme called as TPO, which is your thyroid peroxidase enzyme. This thyroid peroxidase enzyme, it kicks the negative charge of the iodide and converts it into a neutral iodine molecule, that is I2. And when you are kicking off the electron from the molecule, you call it as oxidation. So here's another step in thyroid hormone synthesis, that is oxidation of the iodide ion, which is a very important step in thyroid hormone synthesis. So now once your iodine is oxidized, let's go back a little bit and try and understand the structure of thyroglobulin. Let's say this here is a thyroglobulin molecule. This is a CO minus of the protein and this is NH2 plus. Okay. So now this protein will have amino acids, right? So this thyroglobulin has amino acids called as tyrosine. Okay. So let's say these are all tyrosine molecules on the amino acid thyroglobulin. Now in the previous slide we read about an enzyme called as TPO. Now this TPO has multiple functions. One, it was leading to oxidation of iodide to iodine, correct? Now this TPO will also help this iodine molecule to come and attach to the tyrosine molecules on the thyroglobulin, and this step is what we call as organification of the thyroglobulin. So now let's say that one molecule of iodine gets attached here and one molecule gets attached here. So two molecule here, again two molecule here. Now this is the basic concept. For example, one molecule of iodine on this tyrosine here and two molecules here. Okay. So now let's apply basic concept to it. Two molecules of iodine to one molecule of tyrosine, that is 2 means what? Di, correct? Iodine, that is iodo, attaching to tyrosine, that is tyrosine. So this becomes your DIT, diiodotyrosine. Again, now this is your DIT. Now here only one molecule of iodine is attached to one molecule of tyrosine. So one means mono iodotyrosine. Again, two molecules attached to tyrosine, DIT. So now we have DIT and MIT formed. Now what this TPO, again this TPO will come into action. Now what this TPO will do here? Now this TPO will couple. What will it do? It will couple one DIT with one DIT. That is two molecules here, making T4, which you call as thyroxine, and it will also couple MIT, that is monoiodotyrosine plus DIT, and you'll get, what will you get? T3. And this T3 and T4 are together what we call as thyroid hormones. So here we studied two steps in thyroid hormone. Further two steps. First is organification of thyroglobulin, and then coupling of DIT and MIT to finally form your thyroid hormones. So now, once these thyroid hormones are formed inside the luminal colloid of the thyroid follicles, these are stored within the thyroglobulin molecule. Okay, and each thyroglobulin molecule can contain up to 30 T4 molecules and a few T3 molecules, and these are synthesized in a quantity that can last up to 3 to 4 months. So once these thyroid hormones are formed, these thyroid hormones are still entrapped inside the thyroglobulin molecule, and we cannot excrete them like this directly into the blood. That cannot happen because they are entrapped inside the thyroglobulin molecule. So what happens exactly? So let's understand what happens. These thyroglobulin along with the entrapped thyroid hormones are now endocytosed inside the thyroid follicle from the intraluminal colloid, and this process is called as endocytosis or pinocytosis. Inside the thyroid follicle cells we have an important enzyme called as the lysosomes. These lysosomes are digestive enzymes, and this secrete another enzyme called the proteases. These proteases, they release the thyroid hormone from the thyroglobulin molecule, and this free T3 and T4 are now secreted into the blood. Okay, when this is happening, a small amount of T3 and T4 are again converted to MIT and DIT, they undergo deiodination and form the iodide molecule. And the entire process of iodide into the iodine and formation of thyroid hormone again begins. So now these T3 and T4 are, they act like steroid hormones. Now why do I say that? Because these are not water soluble hormones, okay? So they cannot be in the blood in free form. Therefore, what does that mean? That they need 99% of T3 and T4 get bound to plasma proteins in order to stay in the blood. And what are those plasma proteins? They get bind to thyroxine binding globulin, transthyretin, thyroxine binding prealbumin or albumin. And these hormones are hence released into the tissue very slowly, and in the tissues they get bind to the intracellular proteins and they are stored in the target cell, and in the target cells they are released over a period of days to undergo their actions. So that is how your thyroid hormones are formed and then released into the blood. So let us just quickly revise the steps one by one. So what is the most important basic requirement for thyroid hormone synthesis? That is iodine. You need at least 1 milligram per week of iodine to have proper thyroid hormone synthesis. So what are the steps? First we saw there was formation of thyroglobulin. Then there was trapping of iodide via the sodium iodide symporter into the thyroid follicle. Once the iodide was getting trapped and it was getting released into the intraluminal colloid via pendrin, this iodide or ion was undergoing oxidation to convert into iodine. So the third step is oxidation of the iodide ion. Then there was organification or iodination of thyroglobulin, ultimately leading to coupling of MIT with DIT or DIT with DIT to form T3 and T4. And then these hormones which were stored inside the thyroglobulin are getting released by the process of pinocytosis or endocytosis by the action of lysosomal enzymes, that is proteases, and getting released into the blood. And finally in the blood they get bound to the plasma proteins and get transported to their target tissues where they are stored and released slowly over a period of couple of days. So now, having seen the release of thyroid hormones, how does our body is actually regulating these hormones? The regulation of thyroid hormones is occurring where it all actually started, that is your hypothalamus and interior pituitary. Your hypothalamus and interior pituitary control the rate of thyroid hormone secretion with the help of two important hormones which we studied, that is your thyroid stimulating hormone and your thyrotropin releasing hormone. We know that the thyrotropin releasing hormone was secreted from the paraventricular nucleus in the hypothalamus, which was controlling the secretion of TSH, and TSH was released in the anterior pituitary by the thyrotropes, and the function TSH is a very important hormone. It will increase the proteolysis of thyroglobulin. It will increase the activity of the iodide pump. It will lead to iodination of tyrosine, and it will also control the size and the secretory activity of thyroid cells, thereby making TSH the most important hormone in thyroid hormone regulation. So how is it happening? We know that the TRH is stimulating the anterior pituitary to release TSH, and TSH in turn is leading to the formation of T3 and T4. Now, once our body has enough T3 and T4, the thyroid gland will send a negative feedback to the anterior pituitary and the hypothalamus, telling them that, see, I have enough thyroid hormones in my body, I do not need any more. So it will tell the anterior pituitary not to produce more TSH, and it will tell the hypothalamus not to produce more TRH in order to stop this stimulation. Once the levels of T3 and T4 go down, it will again send the signal that now my levels are low. You can start producing TRH and TSH in order to increase the thyroid hormone synthesis. So now, once T3 and T4 are formed and they have reached the target organs, what will they do? We know that these are metabolic hormones. So, as read earlier, they will increase the basal metabolic rate of the body. How will they do that? They will increase lipolysis, and when lipolysis is increased, they will decrease the fat stores of the body. It will lead to oxidation of free fatty acids and lead to decrease in the levels of cholesterol and triglycerides. In carbohydrate metabolism, they will increase gluconeogenesis and glycogenolysis, which will in turn lead to rapid glucose uptake, increase in the rate of absorption of glucose from the GIT, and increase in insulin secretion. In protein metabolism, they increase protein synthesis and amino acid uptake. In children, they are responsible and very important for the growth of children. In cardiovascular system, they will increase the cardiac output, they will increase the tissue blood flow, they will increase the respiration and the heart rate of, of the patient. Then in children, they are very important for CNS development. This was all about the synthesis of thyroid hormones. I have tried to make it as simplified as possible for you guys. If there are any questions, feel free to ask me in the comments below. So this is me signing off for today. Don't forget to like, share and subscribe to PathDemystified for many more such interesting videos. Bye and see you next time.